Okay, so for our second step into macromolecules, we are looking at lipids. In the form of fats, which is a type of lipid. Um, Lipids also provide insulation from the environment for plants and animals. For example, they help keep animals and plants warm in cold weather. They help keep aquatic birds and mammals dry by forming a protective layer over fur or feathers and using that water repellent hydrophobic property. Lipids are also the building blocks of many hormones and are an important constituent of cell membranes, so they help the cells keep their shapes. Lipids include fats, oils, waxes, phospholipids, and steroids. So there's a few categories of lipids that I'll point out. We'll start with fats and oils. The glycerol, which we see here, anytime you see OL, you know there's going to be hydroxyl groups. OL is a form of alcohol, so you're going to always see an OH when you see OL. So glycerol plus fatty acids, you can see this long chain of carbons. This is a fatty acid. When you combine a glycerol and fatty acid, you get triglycerol. So fatty acids can have a long chain of anywhere from 4 to 36 hydrocarbons. And so that makes the lipids that they produce, the fats and oils that they produce, really kind of big molecules. We sometimes call fats triglycerides. This is an example of one here. And you can hear this term sometimes when people are talking about heart health. They'll test for triglycerides because triglycerides can form cholesterol and plaques in the blood. So there's a few categories of lipids that we'll point out, and we'll start with fats and oils. So fats and oils are made of fatty acids, and those fatty acids can be saturated or unsaturated. Do you remember those terms from the biosynthesis lecture? So why do we call them, how do we distinguish between a saturated and an unsaturated fat? If the fat has even, saturated fats have only single bonds between carbons. So you can see here, there's only single bonds between these carbons. If the fat has even one double bond between carbons, so here's one right here, then it's unsaturated. Okay, so saturated fats, single bonds, unsaturated fats, at least one double bond. And that's only from carbon to carbon. Do you remember which is liquid at room temperature, saturated or unsaturated? The answer is unsaturated. Unsaturated fats are liquid at room temperature. So why are they called saturated and unsaturated? You can see if we look in this saturated fat that each and every single carbon is taken up by a hydrogen. So for the carbon, it's bound to another carbon, but all the other bonds are only for hydrogens. There's no way to add any more hydrogens to any of these carbons. They are saturated with hydrogens. That's why we call it a saturated fat. Unsaturated fats, on the other hand, are not saturated with carbon. So if we look at these double bonds right here, we can see that there is a hydrogen on each one, and there's a carbon, but the other bonds are not taken up by hydrogen. They're actually rebound to the carbon to create a double bond. Plants, some common unsaturated fats are oils. And some common saturated fats are animal fats and dairy fats, like you see in butter. Plants store unsaturated fats in seeds and use them as a source of energy during seedling development. And we've already talked about cis and trans fats in that earlier mini lecture, so I'll skip that part. So how do we test for the presence of fats? In the test for fat digestion, we use a pH indicator, which changes colors as the solution in the test tube go from basic conditions to acidic conditions. And this indicator is called phenol red. So phenol red is red in a basic solution and yellow in an acidic solution. Now remember, fats are made up of long chains of fatty acids. So let me ask you this. If we expect to see red with no fat, and yellow if there's fat, what color do you expect phenol red to be in the presence of fatty acids? Red or yellow? Correct, yellow. Okay, so that does it for this mini lecture for lipids. So the next one up is proteins. Show you a sample set of results. So we can see when there's no fat, the phenol red stays red. If there's a little bit of fat, you start to see the separation of the separation of the fat and the water, or where the fat and the water meet. There's a little bit of fat, so it's a little bit yellow, a little bit red. And if there's fat present, you can see here very clearly that there's fat and mixed in, or trying to mix in with the water. But this is yellow, clearly yellow, so it's fat present.